Understanding Mitosis, created by Brooke Townsend, Shannon Lyons, and Fallon Gallimore. The main purpose of mitosis is the growth and repair of somatic or bodily cells. The result of mitosis is the production of identical diploid daughter cells. Before beginning the process of mitosis, it is important to understand the function and various parts of a chromosome. A chromosome contains genetic information in the form of DNA and can ultimately take on two forms, which are currently displayed. The form displayed on the right side is a result of DNA replication during the S phase of interphase. This chromosome has two sister chromatids, which are bound by a centromere or the kinetochore attachment site. The ends of the chromosomes are called telomeres and ensure that genetic information is not lost during DNA replication. Also, chromosomes have different variations of genes called alleles, which are found at different loci or locations on the chromosome. The phases of mitosis are as follows. First is interphase, which consists of G1S and G2 of the cell cycle, prophase, metaphase, anaphase, and finally, telophase and cytokinesis, which happen simultaneously. Now that we've covered the basics, let's describe and visualize the individual phases of mitosis.
Interphase is composed of two stages of cell growth, known as G1 and G2, as well as the S phase, where DNA synthesis occurs. At this time, the cellular organelles double in number. The DNA appears as uncoiled chromatin.
When a diploid cell enters prophase, the chromosomes in the nucleus are condensed, each chromosome possessing two sister chromatids, which are joined at the centromere. As the centrosomes begin to move to opposite poles, microtubules gradually develop. The microtubules attach to the kinetochore, which assembles on the centromere, forming the mitotic spindle. As this occurs, the nuclear envelope begins to disintegrate.
During metaphase, the fully condensed chromosomes that are attached to the mitotic spindle approach the center of the cell. This center is referred to as the metaphase plate. The sister chromatids bound by chromosome centromeres remain intact.
In anaphase, the microtubules bound to the kinetochore shorten, and the centromeres on each sister chromatid separate. Upon separation, each sister chromatid becomes an individual chromosome. This separation pulls the chromosomes away from the metaphase plate and towards the poles of the mitotic spindle.
Telophase is the final stage of cell division. Here, membranes form around the two groups of chromosomes at opposite ends of the cell to produce the two nuclei of the daughter cells. The spindle disappears and the cytoplasm usually divides, which is a part of cytokinesis.
To summarize, mitosis occurs in order to promote the repair and growth of somatic cells in living organisms. Interphase occurs before the mitotic phases and ensures that the cell has enough DNA in organelles to properly divide. Also, interphase is responsible for synthesizing the specific materials needed for cell division, such as the microtubules. During mitosis, the following phases occur: prophase, metaphase, anaphase, and telophase and cytokinesis. As we have seen, each of these phases have distinct features and play critical roles in proper cell division. Thank you for watching our instructional video on mitosis. We hope you have enjoyed the video and learned much by watching it. The primary reference that we used in making this video is provided here.